begin with the session. I will just share my screen. Okay. So, hello everybody. I would formally like to welcome you to the final class for this particular course, Classics in Neuroscience. So, this is the last time we'll be discussing the previous year questions pertaining to this particular course. And as I did mention last time, what we'll also be studying is certain techniques that have been used and are still pathologically in clinically used to look at the brain and what are the kind of changes that are going on in the brain. Right, so that will be the second half of the session. In the first half, let us quickly begin with the previous year questions for the final time for this particular course. Yeah. So, question number one is what discovery earned Jens Christian Kuhn the Nobel Prize in Chemistry in 1997? Right. So, which was this discovery by this gentleman which actually won him the Nobel Prize in Chemistry in 1997? Right. So, A, was it the influence of cations of enzymes? Or B, the first discovery of an ion transporting enzyme? Or C, the identification of sodium and potassium ions? Or D, the development of active transport membrane mechanisms? Right? So, which one of these do you think is the correct answer? So, James Christian School was actually responsible for identification of these transport channels, right? And what was this transport channel? It was the ion transporting enzyme, actually, not the channel. Also, the ion transporting enzyme was first discovered by Jens Christian Kuhn, right? This is the picture of this gentleman, Jens Christian Kuhn, from the Nobel archives. Uh, and he was actually so Jens Christian Kuhn was from Denmark. He was a Norwegian scientist, right? Uh, Further, what specific enzyme did Scu identify in 1957 that shifted his research focus, right? So, what in the first question, what we had learned was that he had discovered an enzyme which was actually responsible for ion-based transport, right? So the question is, which one of these was that enzyme? Was it the calcium ATPase? Or the sodium potassium pump, or the sodium adenosine triphosphate, or the magnesium ATPase, right? So, which one of this was actually what was discovered by Scope? It was actually the sodium potassium pump, right? So, over here, why it is also a critical question is that it might confuse you because all of these other ones, at least A and D, they have this ATPase written over here or triphosphatase, these kinds of things you have heard. But over here, it is listed as sodium potassium pump, right? Not an enzyme. So that is why it might confuse you. You might rule it out as an answer, but this was actually the correct answer, right? So there are a number of enzymes within this pump actually, and number of proteins within this pump which are at play, which is what he had discovered, right? So this is that complete fact page from the Nobel archives. So this is James C. Scu, as we had described earlier. He has won the Nobel Prize in Chemistry in 1997. He was born in Denmark. That is where he lived all his life, and he died in May of 2018. At the time of death, his affiliation was the same as his affiliation which when he won the award, that is Aarhus University at Denmark. The motivation for his prize was for the discovery of an ion transport enzyme Na plus K plus ATPase, right? So that was the that was what he was accredited with, right? Then question number three, what type of transport mechanism does the sodium potassium pump utilize, right? Even if you have not gone through this week's lectures, you should know this anyway about sodium potassium pump, right? If you are from a biology background, you will know the answer to this. Option A is passive transport. Option B is facilitated diffusion. Option C is osmosis. And option D is active transport. So, which one of these is actually what goes on in sodium potassium pump? It is active transport, right? This. So, if you go into the detail of each one of this, what is passive transport? For instance, there are channels which are always remain open, and we know that if we talk about a cell, the amount of potassium ions inside the cell is more with respect to its concentration outside the cell. On the contrary, the amount of sodium ions inside the cell is less with respect to its concentration outside the cell, right? So naturally, if we follow what will be the gradient of transport, potassium should move outside into the outside of the cell, and sodium should move inside the cell, right? According to its natural gradient. If that is what occurs through certain channels that are open, it is known as passive transport, right? Now these will be leakage channels, right? It will probably not see what change is happening. It will just move into and out of the cell. This third one is osmosis. So osmosis is this transport through a semi-permeable membrane, right? But it is a passive diffusion kind of a thing only across a semi-permeable membrane. So this is actually what is happening over here also. Plasma membrane is in the middle. So that is kind of an osmotic process. What is facilitated diffusion? Facilitated diffusion basically means that if we are talking about just a minute. So what facilitated diffusion basically means that, for instance, something is getting transported out of the cell, but it is also taking something along with it, which is facilitating its transport out of the cell, right? Which is how, say, if glucose also you can take, then you can expel out sodium. If you take ATP, you can take in potassium like that. That kind of a diffusion pattern, right? But over here, what we'll be focusing on is active transport. That is what is the mechanism utilized by the sodium potassium pump. Many of the cell's functions, such as those concerned with nerve impulses, muscular contractions, and digestions, require the concentration of potassium ions inside the cell higher than outside. It than outside it, whereas concentration of sodium ions must be lower inside than outside. So this is the normal physiological condition. And what I'm reading is the concise Nobel Prize work description, right? So it takes a great deal of energy to bring this about, right? Because we have to go against the concentration gradients to maintain more potassium inside and more sodium outside, right? This energy is stored in a special substance, adenosine triphosphate. In 1957, Jens Christian School discovered an enzyme, which was the NAK ATPase, that serves as a sort of biological pump to transport these ions, right? So that was what he discovered. Now, question number four is also from the work of Scu, right? So, J.S. Christian's research, what effect did calcium ions have on the enzyme activity, right? So, yeah, he did talk about sodium and potassium, but he also, along with that, talked about other major ions such as magnesium, calcium, etc. So, what was the result of calcium ions on enzyme activity? One, they enhanced the enzyme activity. Second, they had no effect on the enzyme activity. C, they inhibited the magnesium dependent activity. Or D, they increased the production of ATP. Right? So, which one of these do you think is the correct answer? So, this. answer has to be they basically inhibited the magnesium dependent activity, right? So, if calcium was present in the picture, then what was the effect it was having on enzyme activity? It was inhibiting the magnesium dependent activity of these of the NAK ATPase, right? Then we come to the role of Eric Kandel. Right, Kandel is the person whose original book and the supplemented book Principles of Neural Sciences is what I have been referring the most for this course, right? So, Eric Kandel highlighted the role of synapses in which of these cognitive processes? Was it movement and coordination, or learning and memory, or emotional regulation, or pain perception, right? So, which one of these do you think was the role or the cognitive process which Eric Kandel had highlighted the role of synapses in, right? So, go through that. Mm. Yes. It was actually learning and memory, right? This gentleman is Eric R. Kandel. He won a Nobel Prize in Physiology or Medicine in 2000. He was born in Vienna, Australia, and he was affiliated to Columbia University at USA at the time at which he received his Nobel Prize. The motivation for his prize was for discoveries concerning signal transduction in the nervous system, right? So this signal transduction was through synapses, and there were multiple aspects he has studied regarding this. This is a summary of the work as explained on the Nobel Prize website. The brain is made up of many nerve cells which communicate by sending electrical and chemical signals to each other, right? These signals control our bodies and behavior. Eric Kandel studied how memories are stored by these nerve cells. His breakthrough came in 1970 when he was at New York University studying a marine snail with a simple nervous system. Candle found that the snail learned chemical signals changed the structure of the connections between cells known as synapses, where the signals are sent and received. Right? He went on to show that long-term and short-term memories are formed by different signals. This is true in all animals that learn from mollusks, even up to the man. Right? So that is what he had studied upon. So, in the context of synaptic plasticity, what is meant by homeostatic plasticity, right? So, even if you are aware with the term homeostasis or what does homeostatic by itself mean, you will still be able to answer this, right? So, is it global changes affecting all synapses on a neuron, or changes specific to individual synapses, or mechanisms that enhance neurotransmitter release, or long-term potentiation of memory? Right. So, which one of these is what is meant by homeostatic plasticity? So this is what is an outcome of homeostatic plasticity, which is a global changes affecting all synapses on a neuron, right? But what is this kind of a change going to bring about, right? So, for instance, because we have gone through learning and memory in detail, we have already studied about Hebbian plasticity also, right? Hebbian plasticity, one of the main things it said was that, for instance, if some synapses are firing together, they will build more of kind uh, instance of overlapping between one another. and this synapse will strengthen, and maybe more and more synapses will form at this particular spot so that the signal is strengthened, right? So there is a positive feedback loop due to which there is higher synaptic drive and higher firing rate, right? That is what is meant by Hebbian plasticity. But on the contrary, what is homeostatic plasticity? Homeostatic plasticity, what would be the aim? It would be to attain homeostasis, right? Homeostasis is a basal level of activity, right? So if there is some kind of a low plasticity, that will be rectified so that a basal level activity is achieved, or I'm so sorry. Or if there is high activity, again, that is not suitable probably. So again, it will be stabilized such that a basal level activity is achieved, right? So there is a feedback loop. It could be either a positive or negative feedback loop. If the activity is low, synaptic drive is low, firing rate is low, it is increased so that this dynamic range or this range in the middle, the sweet spot that is reached, even if it is high, right? In some cases, we don't want that. We don't want that firing should go on endlessly, right? That might be true in some cases, such as long-term potentiation, when that particular principle is happening. But if all at all times the activity will be high once, like it has been stimulated, it keeps getting stimulated, then when will the brain rest, or when will it comes to its basal activity, and the next time it has to be fired, then we'll have to go over this high point again to fire, right? So that is something we don't want. That is why we want that after the function has occurred, it should come back to its basal activity range or this dynamic range. So over here, a negative feedback will be achieved if the synaptic drive and firing rate is too high, such that it comes again back to this dynamic range, right? So that is what is homeostatic plasticity. Then we read a little bit about Arvid Carlson, right? So Arvid Carlson was actually another scientist who won the Nobel Prize along with Eric Kandel, right? So what did Arvid Carlson identify as a neurotransmitter in the brain, and what was it presumed, its presumed role before his discovery, right? So was it serotonin, and earlier was it presumed as a precursor for dopamine? Or was it dopamine and earlier it was assumed as a precursor for noradrenaline? Or it was L-DOPA and it was earlier presumed to be a treatment for Alzheimer's? Or it was noradrenaline and it was a neurotransmitter in the Parkinson's disease, right? So, one thing that you can definitely rule out is C, right? L-DOPA is actually a treatment, is a substrate for dopamine. Uh, and it is has been used for as a treatment for Alzheimer's for Parkinson's, not Alzheimer's, right? So this portion over here you can rule out completely. Similarly, noradrenaline is not that much associated to Parkinson's disease as is dopamine, right? So it should be one of A or B, right? So which is the correct answer? It is actually B. Sorry, yeah, this one, right? So what did Arvid Carlson identify as a neurotransmitter in the brain? It was dopamine. And what was his presumed role before his discovery of dopamine as a neurotransmitter? It was actually thought to be a precursor for noradrenaline, right? But it is not just a precursor for noradrenaline, it is actually a neurotransmitter in itself, right? And dopamine is one of the reward-based neurotransmitter also, so it is involved in that machinery also. Moreover, it is a neurotransmitter that is responsible for movements, right, in the body. So this is Arvid Carlson. He again shared the Nobel Prize with Eric Kandel. So he also won a Nobel Prize in Physiology or Medicine in 2000. He was a Swedish scientist who was affiliated to Gothenburg University in Sweden. So I think this is the university where the Nobel is anyway given off also. So the prize motivation for his Nobel Prize was for the discoveries concerning signal transduction in the nervous system. So same as that of Eric Kandel. If we go into the detail of work for Arvid Carlson, a little bit in detail, not in too much of depth, what it says is the body functions of man and animals are controlled by electrical and chemical signals between our cells in our nervous system. So this is something we are aware of, yes. The contacts between these cells are called synapses. Again, we are aware of that. The special substances called neurotransmitters send the signals through the synapses, right? So this is also something now we know, right? Carlson discovered a neurotransmitter called dopamine in the brain and described its role in our ability to move. Right, so dopamine was what he discovered, and he described how it is helping in movement based functions, right? So that is why it is also associated to Parkinson's, which is actually a movement disorder. So in Parkinson's, what happens is there is a loss of dopaminergic neurons, that is dopamine producing neurons, due to which there is loss of activity in these uh, movements of muscles, and or not just activity, loss of control over these movements, right? Rather, this led to a realization that Parkinson's disease is caused by lack of dopamine, as I told you, allowing for the development of drugs for the disease. So one of the drugs that was developed was actually L-Dopa, which was a substrate therapy kind of thing that was given. So if L-Dopa is given, we don't need the actual precursor for dopamine. It is a very, tertiary precursor for dopamine. So it comes almost just before dopamine. So if L-Dopa is present, maybe the neurons which are still active, which have not died, their efficiency to produce dopamine will increase, and the dopamine will still be present so that the function is not hampered much, right? So that was the idea behind it. Then we come to question number 8. In Hubble and Wiesel's experiments, what was the effect of monocular deprivation in kittens on cortical neurons, right? One, neurons maintained normal orientation sensitivity, or most neurons became unresponsive to stimuli from the deprived eye, or the neurons exhibited increased responsiveness to both the eyes, or there was no observable change in the neuronal activity at all. Right. So Hubble and Wiesel were actually scientists who shared the Nobel Prize with Roger Sperry. Last week we had discussed once again Roger Sperry's experiments, how he talked about the connectivity of neurons, and how if the eye is surgically removed, inverted, placed again, the field of view changes, kind of, right? So the inverted picture is not rectified when we talk about its actual image formation or perception in the brain. Rather, the this frog was seeing the fly in the opposite direction as opposed to where it actually was. So that was Roger Sperry's experiment. Hubble and Wiesel also were working on visual cortical stimulation, right? They were mostly working with kittens at the time. And what they saw in this particular case was that if there is a monocular deprivation in kittens, then what will be the effect on cortical neurons? What is monocular deprivation? That means in one of the eye, there will be a deprivation of the signal that is provided, right? So what will happen? Most of the neurons will become unresponsive to stimuli from the deprived eye, right? So this is what is going to happen. So again, this is what I was mentioning. Roger Sperry, David Hubble, Torsten Wiesel, all of them received the Nobel Prize in Physiology or Medicine in 1981. So half of the prize was given to Roger Sperry, and the other half was distributed between David Hubble and Torsten Wiesel, right? Then again, we come to experiment of Hubble and Wiesel. Uh, What did Hubble and Weasel demonstrate about binocular interaction in kittens reared with artificial squint, right? So, what is squint basically? That you might have observed patients or people in real life who do not have the eyes aligned as is the as they are aligned in most of the individuals, right? So, one eye might have a squint, that is, it is aligned a little differently, right? So, what will happen in the binocular interaction in kittens if there is an artificial squint that is created in these kittens? One, both eyes maintain equal influence on the cortical neurons, so it seems very less likely that this will be true. The cells driven by both eyes were abundant. Very few cells were driven by both eyes, showing dominance by one eye only, or cortical neurons became more responsive to auditory stimuli. So, which one of these will have what kind of an effect will this kind of an artificial squint have, right? So, the answer for this is that very few cells were driven by both the eyes now. So, it was seen that one of the eyes was showing more dominance, right? So, if we summarize the work of David Hubble and Torsten Wiesel, this is what it says. Our vision works by the light around us being captured by a large number of sensitive light cells located in the retinas at the back of our eyes, right? So, this we are aware of. This light is converted into signals that are sent to the brain and are converted to visual impressions, right? So, what we are detecting are photons of light. They are getting converted into some kind of a signal that or and some kind of an impression that we can make sense of in our brain, right? Then David Hubble and Torsten Wiesel clarified how this process works during 1960s. In the cerebral cortex, signals are analyzed in sequence by cells with the specific tasks of interpreting contrasts, patterns, and movements, right? They showed how, they also showed that this ability develops in children during the initial period after birth, right? So this is an example wherein you can see this is a normal cat and this is a, strabismic cat which has like squint eye, right? In both the case, both the eyes you are seeing there is kind of a squint, right? And this is the ocular dominance columns that have been mentioned in the in case of the normal versus in the strabismic. And you can see in the strabismic animal, the left and the right eye, right? So this is for the strabismic animal only, both of them, not for the normal and strabismic. It is for the strabismic. So this is the left one, this is the right eye. So you can see that they are more sharply defined and indication of the paucity of the binocular regions, right? So there is more of a distinction between this kind of dominance, right? Also, you can see that this is the case of a normal animal. This is how the cells are distributed. Ocular dominance preference in V1 cells. V1 is that visual cortex area or cortex processing, the visual processing area V1, right? So you can see the distribution pattern in a normal cell, while you can see the distribution pattern in the other cell, right? So if it is strabismic, one eye is showing this kind of a distribution pattern, the other eye is showing this kind of a distribution pattern. So it is very, very different, is what it means, right? Then question number 10 is, what was the key finding from the study on the extent of recovery from visual deprivation in kittens? So once visual deprivation is provided to the kittens, after that, what happens if there is uh a recovery that is tried in this kittens, right? So, A, full recovery of visual functions after deprivation is possible. B, orientation selectivity remained unaffected by deprivation. C is recovery was limited and abnormal response properties persisted. Or D, neurons became more sensitive to the tactile stimuli now that it had been deprived and then recovered. Right? So the correct answer is the recovery was limited and the abnormal response properties persisted, right? So we know that this has something to do with the regenerative capacity also. Kitten is a mammal. There is a very minimal regenerative capacity in these regards in case of mammals. So the recovery was not complete, and this abnormal response that might have stemmed from the visual deprivation, it still persisted, right? So that is the end of the first part of the session, right? So that will be the final previous year questions that we will be discussing for this particular course. And in the second part, what we'll be discussing is the various techniques that are used to study the brain, right? So that is going to be our major focus of discussion. So these techniques are techniques like CT, MRI, PET, other techniques, right? So kinds of MRI, for instance, right? So that is what we'll be discussing. So just give me a moment. I'll just clear my throat. Just a moment. Yeah. So we'll just dive into these techniques that are used to study the brain. So this is one of the charts that you would very commonly see once you talk about techniques or methods of study of cognitive neuroscience. Over here, I have picked up from the book Student's Guide to Cognitive Neuroscience by Jamie Ward. So this is that version of this representation that you might see elsewhere as well. So the methods of cognitive neuroscience can be categorized according to their spatial and temporal resolution, right? So what does that mean? Temporal means something to do with time. Uh, that temporal high temporal resolution will be in milliseconds. Low temporal resolution will be in hours or days, right? And spatial meaning something to do with the space in which it is existing, right? So high spatial resolution will be at the level of a single cell, like a neuron or some part of the cell, such as dendrite or synapse, right? Or low spatial resolution will be at the level of the entire organism, for instance, right? So, when we talk about methods of learning of viewing the brain or viewing certain functions of the brain, right? You can see that there are some methods which are, highly temporally sensitive and spatially sensitive also, such as single cell recording, right? So it is lying over here, meaning it is almost sensitive to the level of a neuron, and it also has sensitivity of capturing even millisecond worth of information. right? So this ideally would be a very good start, right? But at the same time, why it is not heavily used is because it is a highly invasive procedure, right? So there are few things that we need to keep in mind when we are talking about a technique that is used to study the brain. One, what will be an ideal technique to study the brain? A technique which will have a millisecond temporal resolution, a technique that will have a single cell spatial resolution, a technique that will be very minimally invasive, and a technique that will be replicable and affordable, right? So that will be an ideal technique. Unfortunately, that kind of an ideal technique does not exist right now, right? And you can see over here that the single unit recording, which is from a single cell, or multi-unit recording, which will be from multiple cells, maybe, but it will still pick up a few cells. So the spatial resolution will be still higher as compared to, say, an EEG or a PET scan, which will give a whole brain level picture, not a particular cell level picture, right? So over here, spatial resolution will be high, but it will be very highly invasive. So that is why it is still not considered as a good technique, right? Only in very experimental cases will we go into single unit or multi-unit recordings, not in a normal physiological understanding of a patient's brain, right? So we are going to study about these and certain other techniques also associated to these techniques in the future slides, right? Yeah. So one of the major understanding of the brain comes from naturally occurring lesions, right? For instance, when we were discussing about different brain regions and what these brain regions do, that understanding has also mainly come from these naturally occurring lesions. For instance, we have studied about two such examples, such as Broca's region area and Wernicke's area. We know that it is responsible for speech production and speech articulation, right? But how do we know that? Because there were patients in which these specific areas were not there, and either they could not produce words, they could understand everything, they could comprehend everything, but they could not speak it out, or they could not produce words, or they could speak something out, but the articulation of thought was not there, or they could not understand or make sense of what was happening, right? So these are studies that we have gained insights from naturally occurring lesions, right? So that is what this is. So this means studying the effects of brain lesions on behavior and cognition is one of the most established and influential methods in neuroscience, right? In 19th century, the case studies of patients with focal brain damage provided the first evidence that uh, complex cognitive processes, such as those underlying language, have dissociable components that depend on different regions of the brain, right? In brain lesion studies constituted the foundation of cognitive neuroscience that emerged in the mid to late 20th century. Studying Okay. The studies of subject with focal lesions have since contributed to provide fundamental insights in the fields of learning, cognitive control, social behavior, memory, and more. Right. So, two examples for this kind of a study I have given you. Right. So, it was the Wernicke's area and Broca's area discovery. Right. That we have studied also earlier during the course of these lectures. Also, over here, for instance, if you see, this is the brain lesion images from individual patients, right? So this particular patient might have had a brain lesion over here. If this particular one had a lesion over here and this particular one had a lesion over here, right? So if we take the lesions on a standard template, this is what it looks like. And if we try to draw a network of these lesions, that what are the areas associated within this lesion area, it is these lesions, right? And the overlap would kind of give that, okay, all these three patients had a network, affected, which was this network over here, right? So if all of them have some common symptom, it would probably mean that the effect is because of this lesion, right? Or the disruption of that network because of the lesions that are present, right? Then we come to a more realistic version, which is a CT scan, which is something that is very commonly done also. And it is a routine procedure of sorts if you have to look into the brain or at times some other organs also, right? So what is CT? It refers to computer tomography. It refers to a computerized X-ray imaging procedure in which a narrow beam of X-rays is aimed at a patient and quickly rotated around the body, producing signals that are processed by the machine's computer to generate cross-sectional images or slices of the entire body. Right? Over here, we'll be considering only for the brain. So sorry. Yeah. I'll just take cocktails also. Just give me a moment. Okay. So, what is the principle behind CT scan? CT scans are created using a series of X-rays, which are form of radiation on the electromagnetic spectrum, right? So, we have already read about the UV rays, X-rays, infrared, visible light spectrum, etc., right? So the scanner over here emits X-rays towards the patient from a wide variety of angles, and the detectors and scanner measure the difference between the X-rays that are absorbed by the body and X-rays that are transmitted through the body, right? So if it is transmitted through the body, it is going to be less dense. If it is absorbed by the body, it is going to be a denser image, right? So these pictures are then sent to a computer when they're combined to create images of slices or cross sections of the body, right? So what is done is uh, here is an X-ray. Here is a beam of light. It is passed to, say, the brain area over here. One image is captured, right? Then this beam is taken to this angle. Again, X-ray shown. Again, another image is captured, and so on. It goes about 360-degree, capturing all through all these images, and then whatever captured images we have, right, from this plane, then this, then this, then this, then this, and so on for 360 degrees at every interval we will have some image. Then all these images are merged to look at a 3D structure of the brain, right? Or 3D structure of the body. So these images provide more detailed information than typical X-ray images, because typical X-ray images is just a two-dimensional image and a one-plane image, right? These are multiple plane images, because of which we can reconstitute it as a three-dimensional image also, right? They can also show soft tissues, blood vessels, and bones in various parts of the body, right? For this, sometimes we have to do a contrast CT, wherein we enhance how we can detect the soft tissue through X-rays or blood vessels through X-rays, right? So this is what a CT scan looks like for the brain, right? And these are the actual patient's CT scans. I was saying, right, at different planes it will be taken. So it is at different depths the CT scan is there, right? Then we come to an MRI, right? So, MRI or magnetic resonance imaging is a medical imaging technique that uses a magnetic field and a computer generated radio waves. So, over there, what was being used? X-rays. Over here, radio waves to create detailed images of the organs and tissues in your body, right? So, this MRI is arguably the most sophisticated imaging method which is used in clinical medicine as of now. What is the principle behind MRI? These scans work as imaging method due to unique makeup of the human body, right? So we are comprised entirely of cells which contain water, principally made up of hydrogen ions, right? So there are two hydrogen ions in every water molecule. The magnet embedded within the MRI scanner can act on these positively charged hydrogen ions and cause them to spin in an identical manner. Once it spins in an identical manner, by varying the strength and direction of magnetic field, we can change the direction of spin of protons or these positively charged ions, enabling us to build layers of details, right? So when the magnet is switched off, the protons will gradually return to their original state in a process known as precession. Fundamentally, the different tissue types within the body return at different rates, and this it is this that allows us to visualize and differentiate between the different tissues of the body. So, this is the principle, right? So, step one, all protons become aligned with the net magnetization or B0 or B naught. Then in step two, RF excitation pulse is applied to the aligned protons. What it does is aligned proton become excited by RF pulse and flips to the transverse plane, right? So it will go to this plane now, this kind. Then in step four, the pulse will end now and the net magnetization begins to relax and returns to the longitudinal plane. So it had become like this. Now, after relaxation, after the pulse is withdrawn, it will again return back to this kind of an orientation, right? And this is the MRI scan of again different planes of a human brain, right? So you can see the difference between the quality of images that we are getting over here in MRI versus what we were getting in CT scan, right? So this was a CT scan and this is an MRI. So you can see the difference between the kind of, information that we can get via an MRI, which is much more uh, relevant. It is much more, it is just more like you can get more information over here than you can get with a CT scan, right? Then there are various kinds of MRI also that have now come into function. These kinds are basically used to understand how how a particular organ is working or which particular part of an organ is more active. Over here, in case of brain, it is used to determine which brain region is active, at a particular time point, right? So, for instance, it is used in actually patients who are being tested for different scenarios. Maybe Beethoven's music is going on, and then fMRI is done, and certain brain areas are lit up. So those will be more associated to maybe listening to music, comprehending that. And if certain visual stimuli is getting being given, certain other brain region will be more active. So we know that that region is responsible for visual processing and everything, right? So that is what the function will be like. So we'll come to that also. First, let us understand what is fMRI. Yeah. So it is a special type of MRI, which is known as functional MRI of the brain or fMRI. It produces images of blood flow to certain areas of the brain, right? So what it is measuring is the blood flow. Because it is due to the neurovascular coupling, it is thought that if a region of brain is more active, more blood will be supplied to that region. So the flow of blood to that region will be more, and that can be detected via this fMRI, right? And what we presume is wherever this blood flow is more, that is a more active region in the brain, right? Sorry. So functional MRI is a type of MRI scan which shows which areas of your brain are most active. Tracking and comparing that activity to what you were doing at the time can help map your brain activity. It's most often used for planning surgery or similar procedures in the brain, right? So, what is the principle behind this? The principle is dependent on bold imaging, which is blood oxygen level dependent imaging, right? So, what it means is when neurons in a particular region of brain become active, they require more oxygen. Oxygen is carried by blood, and the fMRI detects the relative changes in oxygenated and deoxygenated blood in that particular area, right? So this is suppose a bold activated MRI, right? So you can see that different brain regions are showing activity over here. Maybe it is in response to some different stimulus or some different thought that the person might be having, right? So it can be used for experimental purposes, like I mentioned before, right? To understand which particular area is responsible for which kind of activity. Then we come to PET scan, right? So what is PET? It is positron emission tomography. It is a scan which is an imaging test that can help reveal the metabolic or biochemical function of your tissues or organs. So with fMRI, we were measuring the uh, blood. The level of blood transport that is happening at various regions in the brain. Over here, we are going to test the glucose transport that is happening at these various brain regions, right? Just a moment. Yes. Um, So PET scan uses a radioactive drug, which is known as a tracer, to show both typical and atypical metabolic activity, right? So this is what it looks like. This is a PET scan machine. It looks similar to an MRI or a CT scan machine, right? So over here also there is a rotating scanner, which is going to look at the different body parts. Over here we are looking at the brain itself. And there is a table which will take the patient inside of the scanner and position the correct body part to the scanner, right? Over here, this is what a PET scan looks like. So different colors show different kinds of metabolic activities or glucose uptake regions in the brain, right? So PET scan can often detect the atypical metabolism of the tracer in diseases before the disease shows up on other imaging tests such as computerized tomography and uh, magnetic resonance imaging also, right? So these are basically techniques which look at structural or anatomic changes in the brain, right? So it is possible that before these high level structural or anatomic changes take place, there is some functional change that has already started, right? Much before that. So that can actually be detected much before by a PET scan than maybe any of these techniques. So principle of PET scan involves the use of radioactive tracer called fluorodeoxyglucose. So it is a radioactive form of glucose actually, right? So a small amount of radioactive glucose is injected into a vein, and then the PET scanner takes images of where the glucose is being used in the brain. For instance, this FTG of fluorodeoxyglucose accumulates within malignant cells because of their high rate of glucose metabolism, right? So if there's a glioblastoma or some kind of another brain tumor, neuroblastoma, then there will be more localization of this radioactive glucose at that region, and from that also we can identify that okay, this is the tumor region, right? Then again, in those kinds of studies to understand the function of the brain, also it can be used, right? So this is a resting state of the brain, right? Once the subjects were given music to listen to, and they were given some kind of a language test, these were the brain areas that lit up, right? When they were only given the language, then only these regions lit up, only music, only these regions lit up. So we know that, okay, maybe this side of the brain is more responsible more for the music. Understanding this side of the brain is more responsible for the understanding of language, etc. Right? Then again, what it determines is very high activity over here. This is high activity region, medium activity, low activity, or no activity at all, right? Then we come to just a moment. Yes, just a moment. I'll just. Yes, this then comes TMS or transcranial electromagnetic stimulation, right? It is currently the method of choice to non-invasively induce neural activity in the human brain, right? So sometimes there is some kind of a modulation of neural activity also that is required. So this kind of a modulation or stimulation is normally done also. It is electromagnetic based. It is not like the earlier crude methods wherein electrical stimulation was get being given, right? So this is very less invasive. Over here it is non-invasive only actually. So it is highly used for those purposes. So TMS is a neurophysiological technique that allows the induction of a current in the brain using a magnetic field to pass the scalp and the skull safely and painlessly, right? What is the principle behind TMS? In TMS, a current passes through the coil of copper wire that is encased in plastic and held over the subject's head, right? As the current passes through the coil, it generates that can penetrate the subject's scalp and skull and in turn induce a current in the subject's brain. So this is what it means, right? So there is this TMS. Uh, this is a kind of coil that is given over here. Magnetic current is applied due to the generation of magnetic current. There is this magnetic field that is created due to which a current is induced, which can modulate or affect the brain activity also, right? So that electromagnetism is what is at play over here, right? So TMS is used in clinical neurophysiology to study the nerve fibers that carry the information about movements from the brain cortex to the spinal cord and the muscles also. Technical developments in the devices used for TMS made it possible in the late 1980s to apply TMS in trains of multiple stimuli per second. This form of TMS is called repetitive TMS or RTMS. The repetitive TMS can be used to study how the brain organizes different functions such as language, memory, vision, or attention also, right? So this, for instance, was the brain of a depressed individual, right? So in case of depressed individual, why is it called depressed? Because there is a depression, a depreciation of the activity of the brain, right? But after this magnetic stimulation, you can see the increased level of activity in both the brain regions over here, right? So in depressed patients, the electrical activity in certain areas of the brain decreases, and these are the illustrative images based on these PET scans that was seen, right? So it is a neuromodulation technique, not particularly for imaging. Imaging over here also has been done by PET only, right? Then comes MEG, right? MEG, in MEG, what is the principle? Brain cells or neurons interact with each other by generating tiny electrical voltages, right? So we know that these are the electrical impulses that they are talking about. The flow of electric current produces a magnetic field, just as we talked about in case of TMS also. MEG detectors record and analyze these magnetic fields using sensitive magnetic sensors, right? So the strength of magnetic field from your brain produces is very small. So providers need highly specialized systems to pick up the signals. These sensing systems consist of small, high resolution coils coupled to devices called squids or superconducting quantum interference devices. Right? There are more than 300 of these sensors inside a helmet that fits over your head during the test. So it is also non-invasive. It is on top of your head. Sensors are being placed, and that is where the quantification is getting, happening from, right? The sensors in helmets are specialized computer software, and the specialized computer software work together to detect and record the activity of neurons as you lie still or complete in a series of tasks, such as listening to sounds or looking at pictures, right? So MEG records both the normal and abnormal brain signals every millisecond. The magnetic fields show up on an anatomical image of your brain on the computer screen to map out where specific brain activities originate and when they activate. Right? So this is what MEG images looks like. So this is where the stimulus is being provided, and this is where the recording is happening. Right? Then comes EEG. Again, EEG is something that you might have heard of. It is another common procedure that is generally referred to by the doctors in case they suspect any activity disruption in the brain, right? So, electroencephalography or EEG is a non-invasive and relatively inexpensive method for assessing neurophysiological function that can be used to achieve this goal. So, EEG measures the electrical activity of large, synchronously firing populations of neurons in the brain with electrodes placed on the top of the scalp, right? So this is what the EEG electrodes look like, right? So nowadays there are even caps on which these EEG electrons are placed, and those caps can directly be put on your head, right? So directly they don't need to be placed on the scalp of your uh, head. So during an EEG, electrodes are pasted on your scalp. So that is no longer a requirement. It can be in the form of a cap also that you rest on top of your scalp. These are small metal discs with thin wires, as you can see over here. And this is what the reading from these small electrical discs look like. So during an EEG, electrodes are pasted. Sorry. They detect tiny electrical changes that result from activity of your brain cells. The charges are amplified and appear as a graph on the computer screen that we just saw. So, does an EEG do? It is used to evaluate several types of brain disorders. So, for instance, when epilepsy is present, seizure activity will appear as rapid spiking. So, this is the normal method, right? But in case of seizure, there will be a rapid spike. You can see there is no resting duration in between these spikes at all, right? So, for instance, if you see over here, this is a resting duration, this is a resting duration, this is a resting duration. Similarly, over here also we have some resting durations, right? But over here it is continuously spiking. So those kinds of hyperactivity of the brain causes or hyperexcitability in the brain rather, it causes seizure, right? So that is what we see in that image. People with lesions on their brains, which can result from tumors or strokes, may have very slow EEG waves also, right? The test can also be used to diagnose other disorders that can influence the brain activities. These may include Alzheimer's, certain psychosis, sleep disorders, narcolepsy, etc. Right? And these are the normal EEG brainwave frequencies, right? So understanding these EEG-based brainwave patterns also gives us more information about what is the state of mind of an individual, right? For instance, if we talk about delta waves, which are the slowest waveforms, then these are the waves that an individual normally experiences when they are in deep sleep or dreamless sleep or non rapid eye movement sleep, or even in cases when the individuals are unconscious. I'm sorry. Yeah. Then the higher frequency waves are the theta waves, which are again slow waves, as you can see over here. These are present when a person is in light sleep or deep meditative states or in a recollective state, in a creative state of mind, or they are trying to recall something or they are trying to fantasize about something, right? Again, kind of a dream state, right? Then comes the alpha waves. So this you can see is a little higher frequency than the theta waves also. This kind of a frequency is present generally when an individual is relaxed in light meditative state, the person is creative, or they are conscious, or they are trying to learn something, right? Not very stressful learning, just uh, very superficial level learning. Then comes beta waves, as you can see over here. This again is a even higher frequency waves between 12 to 30 hertz. This is the kind of waves which are more dominant when a person is in normal waking state. Or they are trying to concentrate on something. For instance, right now I'm trying to concentrate on something. Maybe the beta waves are more dominant in my brain, right? When I'm trying to focus on something, or there are five physical senses that are also currently in picture or are involved, right? Then comes gamma waves, which are the highest frequency waves. They generally are active when we are performing a motor function or we are trying to solve a problem or we are highly concentrated into something, right? And if is a continued, hyper excitability or continued gamma wave pattern, that is the kind of situation that we see in case of, or even higher frequency waves, that is what we see in case of epilepsy, right? Then we come to the other technique, which is ERP, right? So what is ERP? ERP means event related potential, right? So some kind of an event has taken place by maybe a group of neurons, and the potential that is developed from that is what is going to be read, right? So it is a change in EEG signal following some type of a stimulus, which is the event, right? So over here, a stimulus is given, and then the EEG is kind of taken, and that is why it is event related potential. This stimulus may be a sensory stimulus, like showing a person a picture of a face or brushing their arm with a paintbrush, right? After these sensory stimuli, the neurons of the brain change in activity, which is then detected by the EEG, right? This change in activity is known as the event related potential, or ERP for short, right? These ERPs are also called a time-locked EEG signal. Since the change in EEG signal is predictable after the event, right? Usually events are measured and detected for up to 1 second or 1,000 milliseconds after the event, right? So those changes are only recorded. So ERPs are more often used for research rather than clinical diagnostics per se. Over here is a setup wherein an auditory or visual stimulus is provided to this subject, and the EEG recording is taken, which amplifies the signal, and this is the kind of outcome that we see. So this is an event-related potential, right? And ERPs are two major features of ERPs are often analyzed. One is latency, which is the time between event and the peak event, right? So how much time it took to actually respond to the stimulus, and the amplitude of the peak, how strongly one responded to that event, right? So broadly speaking, ERPs are subdivided into two categories. One category is the early events, which are those that reach their peak in at around 100 milliseconds. So we are we are recording till a duration of about 1,000 milliseconds. So in the first 100 milliseconds after the event, if the peak is reached, these are the early events. These early events are often characterized as sensory events since they are most prominent after some kind of a sensory activity. Then comes late events, which are those that peak after 100 milliseconds. These are called cognitive events since they are events which correspond most with the complex cognitive functions such as emotional processing. So after maybe some kind of an additional processing has taken place in the brain, then these peaks are occurring. So that is why they are late events, and usually they are corresponding with the cognitive part, right? Then we come to single unit recording, right? You remember I began with single unit recording over there, that it is highly invasive, although it has great spatial and temporal resolution, but still it is highly invasive. That is why it is not generally used. So single unit recording is a method of measuring the electrophysiological responses of a single neuron using a microelectrode system, right? So microelectrode can isolate the spike potentials of a single neuron, which are the signals generated by the neurons when it fires an action potential, right? The most precise recording method today is a single neuron or unit recording using deep needle electrodes, sometimes implanting within the brain, right? So, because it is a recording from a particular cell, so it has to have something that goes and directly measures from that cell. So, deep brain needle electrodes are used over here, right? Inserting a needle into brain is invasive. It is also potentially harmful and it requires surgery, right? It is therefore done only in experimental animals or in humans with medical conditions such as untreatable form of epilepsy, where exploratory surgery is required, right? Where we need to poke into different regions of the brain to identify the focus and then to maybe remove that focus or to modulate it into not showing hyperactivity, right? So this is what the single unit recording looks like. So these are multiple neurons, and the micro needle, it is going within a particular neuron, and it is recording only from this neuron, right? And these are the kind of outputs that we see because it is from a single neuron, right? Then comes multi-unit recording. So it is at a little higher level than a single unit recording, meaning it is less temporally resoluted, less temporal resolution it has as compared to single unit recording, because it is from bunch of neurons which are lying nearby, summated kind of recording is taken, right? By simultaneous recording from neural responses at many different loci at the same time, we can understand the interaction between neurons and thereby gain insight into network properties of neural processing instead of functioning of individual neurons, right? So this is how it is done. So this was the single unit or multi-unit recording. Then comes the single from a single neuron, multi from maybe two, three neurons or five, six neurons lying close by, right? Then comes the local field potential, right, which is from maybe a particular region of the brain. Then comes ECOG, which is from maybe a little higher region of the brain. Then comes MEG and EEG, which is from the surface of the brain, actually, right? So you can see that it is increasingly lesser spatial resolution. So this is most spatially resoluted, this is least spatially resoluted. And so spatial resolution is high, low, but invasiveness over here is high and over here it is low, right? And the number of neurons that we are able to record is a single neuron over here to maybe more than hundreds of neurons over here, right? Then I think this is a final technique that I have kept, which is optogenetics, right? So optogenetics, again, it is not just for exploration, but also for modulation, right? So it is a fairly new technique that has come into picture in the past 1 or 2 decades. So optogenetics is a biological technique used to control the activity of neurons or other cells types with light, right? So opto over here means light, and it is basically to designed to control the activity of neurons, right? It is achieved by expression of light sensitive ion channels, pumps, or enzymes specifically in the target cells. Thereafter, what happens is this optogenetics provides millisecond scale temporal precision, which allows the experimenter to keep pace with the fast biological information processing as well, right? So in systems neuroscience, wherein we are trying to understand how these networks are, connecting to one another, how they are performing well, right? The ability to control the activity of a genetically defined set of neurons has been used to understand their contribution to decision making, learning, fear, memory, mating, addiction, feeding, as well as locomotion, right? In a first medical application of optogenetic technology, the vision was partially restored in a blind patient with a condition which was retinitis pigmentosa. So, in some experimental treatments, it is already trying to be used, right? This is that paper which was for that patient's recovery via optogenetic therapy, right? So it was a partial recovery of visual function in a blind patient after optogenetic therapy. So it was published in Nature Medicine in 2021, right? So that is the end of this part. There are other techniques as well, which are used to study the brain, but most of the, you know, major ones we have covered over here at least, right? So that is what we were aiming to do as well. So I'll just end uh, the screen share. Yes. Uh, so yeah, so that was the aim of our last session. As I told you, that we will go through certain techniques which are used to study the brain, so that that aspect also you will be aware of. So that was it from my end. If there are any further queries, you are free to ask. If not, then because there will not be other classes, you are free to write to me. I will try to respond over mail, right? So other things I might not check, but mail I definitely check. So over mail I'll be able to reply if there is any query you may ask. Um, so if for now there is no other questions. So we'll end the session, and thank you so much for having attended these 12 weeks of lectures, right? This is the final one. So final farewell to all of you. And thank you for your time and patience. I'll be ending the session.